So my privilege today to introduce you Professor Erwin Neher, who won the Nobel Prize in 1991, jointly with Professor Bert Sakmann, discovering the patch clamp technique. So when I was working at the Krennert Institute of Cardiology in Indianapolis, I was working as a PhD student to study the mechanism of sodium channel blocking antiarrhythmic drugs. One day, my supervisor came into the lab and showed a paper telling that it is not fantastic, it is possible to measure now, there is a paper, is it possible to measure current through a single sodium channel. So this was an unbelievable paper that time in '83. In this time, we did not know that this discovery was done a couple of years earlier, actually not in cardiac muscle, but in muscle fibers. And this was one of the very early papers from 1976 when Erwin Neher and Bernd Sakmann presented a single channel recording. So this was a very fundamental paper, and of course that time we were not aware of that this will be awarded with a Nobel Prize a couple of years later. So Erwin Neher, born in Landsberg and grown up in Buchloe, both cities are lovely cities in South Bayern. His father was a company executive in a dairy factory, and the mother was a teacher. And he took the high school studies in a gymnasium in Mindelheim, also a small little town nearby in South Bayern. And during his time, as I could get from his memoirs, that he was interested not only toward living things, but also in physics. So after Abitur at the Maristen College, he took the Technical University of Munich and earned a degree in physics. And as many of us and many scientists also that time, then he took a year in the United States and earned the Master Science degree in the University of Wisconsin in the United States. So returning to Germany, as a young neurophysiologist, started working in the Max Planck Institute of Psychiatry in Munich under the guidance of Dieter Lux. And that was the first time when he met with Bert Sakmann, and they became friends and started to work together. And later on, he moved to Göttingen, the Max Planck Institute in Göttingen, to physical chemistry, and under the at that time his boss was Professor Otto Kreuzfeld, and he allowed the two young scientists, because Bert Sakmann returned from England to Göttingen, to start a new lab in Göttingen for young physiologists. So they started to work cooperating together, and Erwin Neher took 1 year fellowship meanwhile in the Yale University. And after returning, he met with Eva Maria Ruhr, and they married in 1978. Eva was a successful microbiologist, but he stepped back for developing her career to support Erwin Neher and the family. But she was very successful and founded company, a foundation which helps the natural science education of high school students, and this is called X-Lab, and Eva had a program yesterday with the high school teacher here in Szeged as well, helping our program in a great deal. They have 5 children, Richard, Benjamin, Carola, Sigmund, and Margaret. In 1983, he became director of a membrane biophysics department of Max Planck Institute in Göttingen. So before getting the Nobel Prize, he earned several awards between 1977 and 1995 as the Louisa Gross Horwitz Award, Leibniz Award, Gerald Prize, and some others. And of course, many awards and honorary doctor degree after getting the Nobel Prize. So now if we step back in time.
Well, dear colleagues, it's a privilege and pleasure for me to be able to introduce Erwin Neher and Bert Sakmann on this very happy occasion. The events leading up to this year's Nobel Prize started in the late 1960s when the young physicist Erwin Neher and the MD Bert Sakmann decided to register as graduate students at the Max Planck Institute in Munich. So may I ask first Erwin Neher to step forward and give his lecture entitled Ion Channels for Communication Between and Within Cells. Please, Erwin.
So this was the very fundamental, that was the Nobel laureate lecture in the Karolinska in Sweden, and I think that's a nice memory of Professor Neher looking back in time. So the discovery was the patch clamp technique. This made a breakthrough in electrophysiology in general. I just am familiar with the cardiac side. Basically, since the patch clamp technique, we had to redefine almost all the currents, and our understanding regarding the electrophysiology of cardiac cells changed forever, and this became the patch clamp technique was based on several successful drug development projects, and I think was helping to win other Nobel Prizes as well by other scientists. So the principle of the discovery is very briefly the following. If we touch the surface of the cell membrane with a glass pipette, the membrane sticks very well into the glass, forming a very high resistant connection. And for a further suction, we can rupture the membrane, and the interior of the pipette and the interior of the cell will form a common chamber. And with a proper amplifier, we can measure the current through all the channels which is in the surface of the cell, and this mode of the patch clamp technique called whole cell voltage clamping, and this made a very big breakthrough in voltage clamping, because previous voltage clamp technique suffered from a lot of difficulties, so it was impossible, at least in the cardiac field, to measure huge and rapid currents, and this is mostly, I think, successfully used in physiology and especially in pharmacology research. If it is we apply another mode when we touch the surface of the cell and do not rupture it, but gently we pull back the pipette, we can tear out a patch of membrane. This is why it's called patch clamp technique. And through this small, tiny part of a membrane, we can measure the function one single channel, depending how dense the channels are in this patch, maybe 2-3 more channels, and this can be used very elegantly, mostly I think in physiological research. So this discovery was published in several important papers. Actually, I just would highlight the Journal of Physiology and the Nature paper in 1976. They earned, as you can see, many citations, and when they developed further the technique, there was a very important paper published in Flüger's archive, and where the patch clamp technique was described in a very great detail, and this paper from until now earned more than 20,000 citations. So then Erwin Neher worked in Göttingen, and since 2011 he became director of Emeritus Group of the Institute of Biophysical Chemistry and Membrane Biophysics in Göttingen, Germany. He was also involved in drug discovery research, having an institute in Macau, China, to apply this technique on pharmacological research. His current research interest, and back for many days from now, and just I picked up two papers just by chance. The excitosis, mediate neurotransmitter release, synaptic short-term plasticity, and developmental aspect of the synaptic transmission. So, but Erwin Neher has also a connection with Szeged, with our city and university. He visited Szeged twice before, in 2015 and 2019, helping our program in a great deal, discussing with the students visiting high school, Radnóti High School in Szeged and Németh László High School in Hódmezővásárhely. And I think we can be very thankful to him, devoting his very valuable time to go here, and he will present his latest research for the students. And I would like to ask Professor Neher to deliver his lecture of today.